tira, kua taimai nei kiko nei, ko Shane, ko Hore, ko Ian Mitchell, tēnā koutou, tuatahi, tuaru, tuatoru. I ngā tangata kua taimai nei kiko nei, o tēnei hui, tēnā koutou, tēnā tātou katoa. Ko wai au, ko Phil Wilcox tōku ingo, no rāki rākai pāka, ngā te kahungunu, ngā te runga mai wahine, oku iwi, ngā te rākatoa, ngā te apatu, me te wharirau a te tākinga, oku hapu. He kai pūtau ahau e a Sion. Me te whariwānaka o Otago. And I'd like to just dedicate my talk today to a good friend and colleague who passed away 2 days ago, Kath McBride. So just a little bit about myself, my professional background this time, my qualifications. I started off as, and you've heard a lot about the Society of the, what I call the Society of the Green Triangle, the New Zealand Forest Service. I worked for them for a number of years, then I went into research. I have a PhD in disease, and my thesis work was in disease resistance, which I did in the United States. I'm a senior scientist with Sion, but also a senior research fellow with the University of Otago. You can all read those words up there. Basically, my roles and my scientific roles have been around trying to develop modern genomic technologies for application in a whole host of areas, primarily forestry, but more recently I'm working with human geneticists in diseases that typically affect Māori and Pacific populations. I also have a background in Māori and science engagement. I have an iwi role, one of the iwi I come from, Rākai Pāka. We have a health and ancestry study which involves a fair amount of genomic science in that study, so I have a technical advisory role with that iwi. It's not a paid role, but I do get good access to good power diving spots. And both in the Sion work that I'm doing, as well as the University of Otago and University of Waikato work, is around developing appropriate processes for engaging with Māori communities to ensure those communities can actually benefit from these technologies. And I'll talk a little bit about that in this talk today. Now, the previous speaker raised the importance of genetic resistance, and so my talk today is really about what are the prospects for genetic resistance in Cody. Now, Ian gave a very nice summary this morning about the symptoms of Cody dieback disease. It's probably not a good use of my time to go through those again. But one thing I will take note of is that in the infection experiments with seedlings done by Ross Beaver, and Stan Belgard, more recently, they've been able to kill every seedling that they've screened. And it also affects trees across the age class distribution. So there's a suggestion, well, not just a suggestion, but a strong hint of a severe genetic vulnerability within Cody. What I'm going to say today is that it might not actually be that bad. We should expect to see genetic resistance in Cody. That resistance will be uncommon, and it is likely to be randomly dispersed, and I'll say why that is. But also give you some examples of where genetic resistance has been very useful in breeding programs of other species overseas, as well as some in New Zealand. And that a successful breeding program will involve multiple components, and I'll talk about those. And also some update on where we're going with the development of screening technologies. And then I'll touch on how we might be able to utilise genetic resistance. And I think that there are not just limited but large opportunities with heritable resistance. So firstly, why should we expect to see resistance to an introduced pathogen? Well, first of all, and you've heard mention of it this morning from Graham, the appearance of asymptomatic trees in amongst diseased trees. Now that in itself is not proof of genetic resistance, but what we do see is that it happens quite frequently. So the previous slide I showed was from Huia in the Waitakere, I understand, with work done by Monique Wheat, who's Bruce Burns' former master's student. Bruce and others have gone on to map the location of asymptomatic trees in amongst diseased trees on other sites as well. So the point there is that Huia site is not unique in having asymptomatic trees occurring on in amongst diseased trees. So, but what I want to do now is just really talk about, well, what's the experience overseas in native species with introduced pathogens of forest tree species. And the point here is that the resistance is the norm. Now there's a number of examples: white pine blister rust to sugar pine and white pine, American chestnut blight to resistance to the causal agent, which is a Cryphonectria parasitica. And most importantly, I think for our discussions today is resistance in Port Orford cedar to Phytophthora littoralis. Now, Pinus radiata is also indigenous to the United States. In this environment in New Zealand and in Chile as well, we see resistance to a range of pathogens, including Dothostroma needlebite, Fusarium, which is pine pitch canker, and more recently, resistance was shown to Phytophthora pluvialis by my science leader and colleague Heidi Dungy, who's here today. Phytophthora cinnamomi resistance has been shown in Western Australia, and also Phytophthora cinnamomi resistance occurs in Jarrah, which is Eucalyptus marginata, which is an indigenous eucalypt to Western and other parts of Australia. Nari Williams, who's here today, and Peter Scott both have a lot of experience working with Phytophthora cinnamomi. Cupressus macrocarpa, which is also an indigenous species to the Western United States, has resistance to cypress canker. So these are just a few examples. So the second point I want to make is that resistance is likely to be uncommon and randomly dispersed. Okay, so one example, white pine blister rust. It's caused by Crenatium rubicula, which was made its way into the United States sometime in the early 1900s. Hasn't been enough time for the disease for resistance to naturally arise and then spread through the population. But nonetheless, what you're looking at here, you see all these triangles. Most of them are black triangles. This is, I should point out that this is west coast of the United States, or part of the west coast of the United States. These black triangles and the red triangles are all the location of trees that are asymptomatic, i.e. they haven't got white pine blister rust in areas where there is blister rust. And what the guys did there is that they took seed from those trees and they screened seedlings that were raised from that seed and then assessed the rust resistance in each one of those families. Now, where you see the little red triangles is where you see resistance. So that resistance is scattered. It is uncommon. It's not quite rare, but it is uncommon. And it's throughout the distribution. And I'll just repeat the point that I made before. That distribution cannot be explained by a mutation arising in the host to confer resistance and then spreading. This to me looks like it existed well before the disease actually occurred. Why that is, I don't have time to talk about, but I'm willing to bore you to death about what my theories are afterwards. Most importantly for us, Phytophthora littoralis resistance in Port Orford cedar. Now, Port Orford cedar is another species endogenous species to the western United States. We're looking at the west coast of the United States in that picture there, and the distribution of Port Orford cedar. Basically, the disease Phytophthora littoratus was first discovered in 1920, and it's a soil-borne pathogen just like PTA. And resistance was first reported in 1989. A breeding program was begun by the USDA Forest Service in 1997 under the direction of a colleague of ours, Richard Snesko. They have screened over 1,260 seed lots taken from asymptomatic trees in areas where there has been tree deaths. So asymptomatic trees amongst dead and dying trees, and one of the examples is up here. You might not be able to see it very well, but these are dead spars in a green tree and amongst brown and dying trees. Okay, so what you're looking at here is the same kind of thing. We've got a distribution of resistance where seed lots were taken from trees that were asymptomatic, like the one I showed in the previous slide, and then basically the distribution is shown on this graph. On this map here, you have purple, which are the susceptible trees, susceptible seedlings, but the resistant ones were where the seedlings showed high levels of resistance. And so we were able to see once again that same kind of pattern. Resistance seems to be more frequent with this pathogen, but it is nonetheless spread throughout the range, the entire range. This isn't actually the entire range, but if you look at the entire range, you'll see the same pattern. Throughout the entire range, you'll see resistance distributed. Okay, and this is just this little slide here just to show San Francisco's down here, Seattle's up there, and this is the area that we're looking at. And it's the point of the point of stating that is that we're pretty much the same kind of temperate zone, northerly latitude as we are in southerly latitudes. So the Port Orford cedar story is a very encouraging story, and one that I believe that is an interesting exemplar of how a breeding program can be utilised. So my third point is that such naturally occurring resistance to exotic pathogens has been very useful in other species, and I've listed a whole set of examples up there. Dothostroma resistance in radiata pine. You can buy Dothostroma resistant material. You can buy cypress canker resistant material that was developed by Sion colleagues a number of years ago. In the United States, there is chestnut blight resistance now being put into breeding programs. There's been a white pine blister rust breeding program that's been quite successful for a number of years, and you can buy these so-called Liberty elms that are resistant to Dutch elm disease, elm trees that are resistant to Dutch elm disease. So here's an example from Port Orford cedar screening. Basically, we've got a susceptible seed lot where 100% of the seedlings have disease, which is similar to what we're seeing in Cody, but the offspring of 2 trees that were shown to be resistant in the screening that they've been doing have less than 50% mortality. So that's the kind of outcomes that can be achieved from resistance breeding. So what will a successful resistance breeding program involve? The first thing is the development of an effective screening system. Second thing is once we have that, we're going to have to, in my view, because of that unpredictable, random and dispersed nature of resistance, is sample from pretty much that entire distribution of Cody. Okay, so that means a coordinated effort across Cody lands. And it also means, in my view, and I'm sure there are others here who agree, that there needs to be extensive iwi consultation about that regarding the use of this genetic variation and the application of the certain technologies that we would think have some value and benefit. Y262 has not been The Waitatū Treaty, Flora and Fauna Treaty claim has not been settled. And that's a pity, because if we had, then we would have an environment with which to conduct this research. So that appropriate environment needs to be established via a lot of discussions, as Matua Hori alluded to this morning. Some other science stuff that needs to be improved. Seed production and propagation methods need to be improved so that we can accelerate the identification and the deployment of resistance. And I would propose too that we need to give consideration to the development of DNA fingerprinting technologies and to a full genome sequence of Kauri. And so why do I think that, and how that might actually help accelerate the identification and deployment of resistance? So we've got our disease stand. We've got asymptomatic and symptomatic trees. Basically, if you extract the DNA, assess the trees, extract the DNA from multiple sites, and then you fingerprint those trees. The trees that are most, if resistance is hereditary, if it's genetic, then trees that are most likely to be, the trees that have the most similar DNA fingerprinting pattern and are most likely to be resistant, well, they're assessed that are most likely to be resistant, you'll get a correlation between the DNA fingerprinting pattern and resistance. And so that means that you have some confidence in which trees are truly resistant trees without the need to go and screen. And so it cuts out that screening stage. And that's one of the ways that this technology can speed things up. By identifying those trees that are truly resistant a lot faster. You can take seed from those or intermate those trees, plant them out in nurseries, and then out and propagate them, and then out into the forests. So where are we at? Peter Scott will give a presentation soon about work we're doing in collaboration with the Bioprotection Corps at Lincoln, essentially where we're at with developing the screening systems. So we've only really made a start in this particular area. So if we find resistance, what might be the opportunities for and how we might utilise that resistance in combating this disease? Well, first of all, to enhance resistance around iconic trees and ngahere that are particularly important, and some of those isolated pieces on private land where people wish to essentially have a barrier to resistance that is cheap to implement. And there's a particular example within Phytophthora cinnamomi resistant, deployment of Phytophthora cinnamomi resistant Jara, where that has been used as one of many management strategies. My colleague from Scion, Lania Holt, will talk about the value proposition for managed kauri forests, and it looks quite exciting, but you don't have a reasonable value proposition unless you can manage the disease. And tree species of economic value that do suffer from disease always need to have a breeding program which part of it is dedicated to combating the disease. Breeding can increase growth rates by 10 to 15% per generation, as well as increasing disease resistance. And I think also something that we need to give due consideration to is the economic development opportunity. The silver lining on this particular PTA cloud is that there may well be opportunities, economic development opportunities for iwi. Again, Y262 has not been settled, but this may be a wonderful opportunity for the government to do so. So key messages, once again, we should expect genetic resistance. That resistance is likely to be uncommon and randomly dispersed. Nonetheless, it's going to be quite useful. The breeding program will have a range of components to it, and the development of screening techniques is underway. And I talked about some of the opportunities, but I think the big picture around the opportunities is maybe PTA needs to be looked at from an opportunity point of view, and that achieving the goals of cultural, heritage, environment, conservation, education and production are not mutually exclusive goals. In this particular case, all of those goals I think are achievable, and that genetic resistance is just one of the many tools to do so. And finally, just to leave you with a whakatauki, which I think is particularly appropriate, te manu e kai ana i te mero nō nā te rangatiratanga o te ngāhere, ahakoa. Te manu e kai ana i te mātauranga nō nā te rangatiratanga o te ao. And with that, I'm done. Thank you.